A warm welcome to all of you at the first day of the AMR conference 2023 in Basel. This workshop has already a very long tradition and it is supported by the educational initiative of the Repair Fund, GARD-P, CARB-X, Wellcome Trust, JPI AMR, and the AMR Action Fund. I would also like to acknowledge Lloyd Payne for helping to design this workshop. This year's program focuses on a very timely topic. As in other fields of our life, artificial intelligence and machine learning strategies are very prominent. And we have three speakers here who are covering a broad range of perspectives and also personal experience related to drug discovery. With this, I will welcome our first speaker. It's David Zhang. He's a computational biologist working at Hoffmann-La Roche, where he develops new experimental and computational methods to identify novel drug candidates and to understand their efficacy and safety profile. Please, David.
Thank you, Ursula, for the introduction, and welcome everyone. My name is David, and today my talk has a title, Finding Hope in a Hopeless Time. And this may sound very unusual for a time that many people celebrate as a springtime for artificial intelligence. And I will try to explain my rationale for this. And before I start, I want to have a quick check with you. Who will identify yourself as a biologist/chemist? Raise your hand. Thank you. Who of you will identify as a medical doctor and or clinical microbiologist? Welcome, thank you. And who will identify you rather with artificial intelligence, machine learning, and next generation data analysis? Also a few, good, cool, thank you. So we have a very good representative audience, which I'm very happy and thankful for. So I think we have a wide range of expertise that can criticize my mistakes and ignorance, so please do so. So my topic will focus on how predictive modeling and data analytics shifts our perspective about antimicrobial drug discovery. So in the coming 20 minutes, I want to introduce three things. First, I want to introduce predictive data modeling and data analytics in drug discovery, which I define it as integration of knowledge, experiments, and machine intelligence to establish causality. Second, I give you three pieces of case studies of PMDA in antibacterial drug discovery, both in our company as well as in academic research, including imaging, molecular phenotyping, and single-cell data analysis. And last but not least, I want to explain a few challenges and prospects that I see now. So what do we mean by predictive modeling? You have heard a lot about artificial intelligence, machine learning, data analytics. And all of this actually have a few implicit assumptions. For example, you can distinct or you can separate intelligence from body. For example, you can actually use computational power to solve problems. For example, you can use a machine to mimic or even to create intelligence. And my personal standing is that recall that when we are dealing with microbiome, we are dealing with bugs, we are dealing with a real type of intelligence, which arguably is stronger than human intelligence. So this, I call it microbial intelligence. Namely, they do evolution, they constantly change themselves, and they can actually adapt to environment. And this is actually so far the artificial intelligence cannot yet do. But it's not totally hopeless. And that is what I meant by finding hope in a hopeless time. And this is, by the way, from a song that I like very much by a human, not yet an artificial intelligence player, called Joshua Hislop. And To define the predictive modeling, there are 5 hallmarks. First of all is human intelligence. And second is machine intelligence. So this is what I now will talk about: machine learning models, artificial intelligence models, data analytics. And the third, experiments and observations. Models exist in our mind, and this, especially a very big part of this model, are called causality. And causality, in a layman language, if I try, is to ask what-if questions. What are you now doing if you had not signed up this workshop? And last but not least, it's an iteration. It's a process. It's never a one-ending story. We get a big model release and everyone is using it. That's perfect. That is the end of the story. No. We are dealing with real bugs that go evolution every second. And what's the relationship between this? Well, human would have models. And the models, actually part of the model is about causal, about what causes others to happen. And we do science by actually overlap in the interface between our models and reality. What we can do experiments, and we can use observations. For example, I just did an experiment when I started. Who are you coming from? Field X. So this is actually an experiment. At the same time, it's an observation. And this observation experiment comes from my question setting. Namely, I'm curious, what kind of audience do I have today, and what concept I need to explain more and explain less? I could have asked a different set of questions. I could have asked, where do you come from? I could ask, what did you eat today? But for my purpose, and this purpose comes from human intelligence, I want to ask specific questions and set up the experiments. And at the same time, nowadays I can use machine. I could have not asked the questions. I could send you a QR code and all of you scan it, and I can get the answer much more precise. But all this actually flows to the interface between the models and reality. And that is one very strong assumption, that we do not deal with reality directly. We always deal with reality with models and experiments. And what we define science translates between models and experiments with creativity, ideas, and even irrationality. And this is my personal definition of what we do drug discovery today. And in my opinion, it's more broad than artificial intelligence, though this machine intelligence, what I call them, is part of our arsenal to target bugs. So in the coming 15 minutes, I would like to focus mainly on 2 types of data now you can get, both from your lab as well as from public domain, that may help you with your work. Especially so many people come from a biological/medicinal background, a chemistry background. I think I will not go to the biology too much, but rather focusing on the data aspect and on what can we do with these technologies. So essentially, I want to introduce you to 2 types of data. One is Omics data, the other is morphology or cytology. The Omics data essentially is what we call Omics. It's a pseudo-Greek word. It means everything of a particular kind. In real life, actually, we don't decompose ourselves into these atom-like elements. But for simplicity, if you imagine the middle, I show you a biological pathway that is actually has a pretty complex mode. You have actually induction and then regulation and then a feedback loop even. What Omics does is like lenses. It sheds using different technologies on this network, for example, using NMR, nuclear magnetic resonance, using mass spectrometry, and using RNA sequencing. So when you observe with these technologies, you can observe transcriptomics, proteomics, metabolomics. That means this provides you a particular perspective looking at biological systems. Do not forget that these are not biological systems per se. It's as if I'm now asking you to stand and talk with each other. I can work from different angles and look at you, but I'm not studying you yourself. I'm just studying the components. How do they form each other? The biological network, so to say, can be viewed from each individual atom level. Also, they can be viewed on a much higher level of phenotype, and this is known as morphology or cytology in the bacteria case. This is actually very well known in day one when Hook looked at under microscopy, they realized that, okay, different things have different forms. And now with the advancement of omics and imaging technology and machine learning, we have now gone through a transition of how do we see things. Before we think that, okay, this is biology. Now we think that cell identity and cell state can be represented as high-dimensional data. So again, this is a representation. It's not the real biological thing. But you can use this data for many purposes. For example, screening. And here I show you the principles of one of this technology called cell painting. Essentially, you have specific dyes for different parts of the cell, nucleus, endoplasmic reticulum, nuclei, actin, mitochondria, and segment. And if you merge them together, you can segment them very well. And by doing this, what you can achieve is that you can use genetic and chemical perturbations, do experiments in multi-well plates, use microscopic imaging, and back to artificial intelligence, one of the really strong points of machine intelligence is that it can go through large amount of data and find correlations. And you can use image analysis, and finally you get this morphological profile. Namely, you have cells in the row, you have the features, how much DNA, how much RNA, how much protein, how much lipid, etc., one concatenated after another, and you can make very high throughput screening with this. So this is really one-on-one of phenotypic screening with bugs. And this piece of work has been done since more than 10 years, actually probably much longer, but since publication is about 11 years now, where the group have shown that it's a publication in PNAS 2011, bacterial cytological profiling identifies cellular pathways targeted by antibacterial molecules. On the left side, you see that there are different images of drugs, for example, tetracycline, which belongs to the class of protein translation inhibitor with P1. So the group, what they did, they tested many drugs of many classes, and then they built a simple machine learning model. For example, they continue use another RNA translation inhibitor, rifamycin, and then triclosan, etc. They use 5 classes: protein, RNA, DNA, lipid biosynthesis, and cell wall synthesis inhibitors. And they concatenated these features together and run what we call a PCA, principal component analysis, essentially just a way to look at where are the maximum difference between these drugs. And it's told and sold, they are well separated. And this bears a lot of interest, among others, also in Roche. A few years ago, my colleagues, Hoffmann and other colleagues in the group, they did a very similar exercise. They have individual parameters now in the row and the column. We have different drugs, including DMSO. And we could do a dimensional reduction, for which PACA is one type of method, that we can see that the compounds clustered by their MOA. And then we use reference compounds, and then, so essentially for those, we know how they work against the bugs. And then we go to test compounds. We could verify that actually, even we have hidden the labels of MOA of the test compound, we can recover them. And this is one of the concrete applications of machine learning drug discovery. And what do we learn? We learn that machine learning can learn morphological features of the bugs upon treatment that are reflecting the MOA. But what we cannot learn is that actually why and how do you come to these molecules at all? And how to come to this new chemistry is a difficult question. And one is actually how do you penetrate the cell wall, for example. The other question is actually how do you tell whether a drug is safe or not, is likely to be safe to be used in clinics. And for that, we have developed, among others, a platform called molecular phenotyping, which we can use for many different purposes, among others, for antimicrobial and antivirals. So the idea is that whatever therapeutic candidates, you can have small molecule, large molecule, antisense oligonucleotides. And you can use human in vitro disease models, cell lines, primary cells, IPS, so induced pluripotent stem cell derived cells, or advanced models like organoid. And we use very early time point because from the system biology point of view, the early time point shows the initial primary effect of the target engagement and the primary signaling induction. And we have predefined about 1,000 pathway reporter genes, so these are human genes that we selected based on human intelligence and machine intelligence. We know that these are specific regulated when certain pathways are regulated, for example, mitochondrial toxicity or lipid metabolism. And we use sequencing technology to make the readout. And the technology is published, and as we know, many colleague groups around the world, both academia as well as companies, also use this technology. Finally, you get essentially a matrix of how molecules induce human pathways. So this is a dendrogram showing the 154 pathways that we could detect using the molecular phenotyping platform. And if you ever ask me to draw this with my hand, I will say no. This is definitely a job for machines. At the same time, though, the knowledge comes from humans. So we ask biologists, chemists, toxicologists to have a look at them and let us know what they think about these pathways. So we have done a study, or we have done many studies, but this study is one of the examples that I want to show you today. Namely, we have a Roche compound, which is a Roche antibiotics, and we have daptomycin and colistin. The reason is actually we saw in vivo toxicity of this Roche compound, and we want to understand why. And we use 3 types of cells, in iPS-derived human cardiomyocytes, primary human skeletal muscle cells, and human embryonic kidney cells. And we use molecular phenotyping. So finally, we see that actually the Roche compound on the total expression level looks very much like daptomycin and less about colistin. And we see that on many pathways level, also we see the Roche compound is more like daptomycin than colistin, though the chemical structure would have told us the other way around. So this is actually the added value. Essentially, we can use Roche molecular phenotyping to characterize the compound mode of action or safety with reference compounds. And the last example I want to give is a recent progress about single-cell RNA sequencing. So instead of sequencing a bunch of cells, you can also use sequencing to sequence individual cells, even the bugs. And this is one of the new technology called backdrop. Essentially, it consists of 3 steps. First, you delete or deplete RNA and DNA in the bug. And then you perform reverse translation to put RNA back to DNA. And finally, you use droplet platform to generate droplets and sequence these bugs. And the authors of this paper, which published in Cell 2023, so about a few weeks ago, they used a clinical isolate. They could identify the different species, including both gram-negative and gram-positive bugs. And also they treated the Klebsiella with different antibiotics. They could see that these antibiotics separate the bugs into each other. So this plot we call the UMAP plot. What is a UMAP plot? Each dot is a bacterial cell. And if 2 cells are very near to each other, there's a likelihood that these 2 cells have very similar gene expression profile. Namely, they have very similar composition of expression of the genes. And now you see that actually upon treatment, the 3 drugs actually drive 3 different expression profiles. And they went further into looking at meropenem, and they identified a very heterogeneous cell response, which is not surprising. We know that actually, coming back to what I told before, we are dealing with real intelligence. The bugs are not sitting there waiting for that. They actually have different strategies. And their strategy is actually by population. They divide into different cellular subtypes. And among others, there is a special cluster called CSPD. For those who studied microbiology, you may recognize this. It's cold shock protein D. And it's a surprising thing is actually that the author went on testing that the CSPD cluster are enriched of resistant developing bugs. And this could not have ever been struck to us if we don't have this technology. And this is a very concrete application of machine learning and artificial intelligence, namely go through large number of data and identify what are the potential causes of novel resistance. So with that, I finish my talk. I see 3 challenges and potential prospects. I said, I think given the a huge number of potential resistant developing mechanisms, and given that much of the investment today go to artificial intelligence, I'm really thinking that we are in a hopeless time for antibiotic research. And one of the reasons is that we have to find gram-negative bugs. I think 2 things are important there. One is to have new chemistry to penetrate gram-negative cell walls. The other is learning from failures is as important as from successes. The second challenge is actually that we still know too little about how drugs work. And there, I think phenotypic drug discovery is important. Molecular phenotypic drug discovery is even more. Namely, we have to understand on a molecular level how these drugs work and how do they induce both pharmacology and toxicology. And one difficulty is to dissecting pharmacology from toxicology, but there in antibiotics, we have a very good chance to do this because the primary target is seldomly human gene. And we need better preclinical models with high predictivity for clinical outcome. I think so far, majority of the development in most of the companies are still done with rather artificial models. Artificial models give artificial results. And the last thing I want to mention is that acting against resistance from day 0. One possibility is to use Omics enhanced resistance detection and understanding. I think the single cell study provides one look into what we may do routinely in the coming years. And last but not least, I want to thank the colleagues, especially Ursula, for inviting me, and other colleagues who will speak after me. And because I think such kind of pre-competitive knowledge sharing is essential. With that, I want to share what Roche asked us to do, doing now what patients need next, and thank you for your attention.
Thank you, David. I couldn't agree more that we need more knowledge and pre-competitive knowledge sharing. With this, I would like to invite our second speaker. It's Nikolaus Stiefl. He's a data scientist working at Novartis. He's a trained pharmacist, but switched to chemoinformatics, machine learning, and molecular modeling early in his career. He's part of multiple small molecule drug discovery projects with delivering a range of discovery candidates. And he will talk about drug discovery at the interface between computer-aided drug discovery, chem informatics, and research informatics. Please, Nick.
Thanks, Ursula. Thanks for the kind introduction. So let me see how this works. Thanks. Right. Okay. Thanks, everyone. Thanks for inviting me. It was short notice, so I hope I get the topic right. It was a great talk. Thanks a lot, David, because I will actually pick you up on some of the pieces in what I'm going to present. So that's good. That's the only formula I'm going to use today, but I think it's an important one because it's essentially showing what you need to understand about a model. And I'll be talking quite a bit about machine learning and models and maybe a little bit about the established CAD technologies. So what we're typically doing with this thing is that, we have a y, which can be described by a function of a delta in x and an error. Probably brings you back to your university days. It's actually quite simple. And this is, if you want, the more established, old way of doing machine learning. And afterwards, I talk about a lot about these new things a little bit as well. So the y is the thing you actually want to predict. Can you predict activity? Can be a permeation? Can be whatever. The function is your machine learning model. The delta of your x is essentially the thing that you want to predict and the differences between this. And that's important to keep in mind. When you build a model, what the model can learn is a difference in what you have. It will not be able to come up with something completely new from nothing, right? Just keep that in mind. That's really important. And then the error. I think the error is actually really interesting. The error is actually one of the most interesting pieces in all of this because it shows you where the golden nuggets are sitting in your data set. Okay, so it's great, the formula is perfect if it only would work. The thing is that in the real world, we have really different sets of data. We have small and sparse data, that's one of the caveats. And this is from a study that Eric Martin has done, and come back to him later as well. Roughly 14,000 essays, and when you actually look at this through our internal databases, we have 0.1% of the data that's only filled. So it's super, super sparse. You cannot really learn on everything just like this. You have to be smart, and Eric was that, so I will talk about this in a second. A lot of the data is censored. That means it has like a larger than or less than, so you cannot really do a numeric approximation of something. You know, what is two compounds? One is bigger than 30, the other one's also bigger than 30, but one might be 31, and the other one might be, 5,000. You cannot approximate this. There are some statistical methods to do this, but it's not super straightforward. And then you see those distributions. No, you don't see the distributions. How do you go back? Okay. No laser pointer. Okay, so what you see there is essentially you very often have at the extremes those bimodal distributions. So if you see that kind of thing and you want to model this with a model that tells you a specific number at the end, for example, that's very hard to do because the model will be biased. So in these cases, you need to go again to specific types of models. Come back to this in a second. For the small data, it sounds like, yes, we have a lot of data, and yes, we have a lot of data, but very often we only have very small data sets. You see 50% of our data is less than 500 data points, and most of those are clustered in a very close space, so you can actually really do a lot with this. Data shifting is another big problem that you need to keep in mind. So this is Proteus IC50 data panel, and what you can see is from 2005 to 2009, you had a really nice distribution between actives and inactives. 2009 to 2013, suddenly only inactives. So you can say, like, what the hell is going on there? What's happening is that in the end here at the 2009, you have that shift. Before it was a project assay and then became a panel assay. So that means only other projects would use it for checking if something is wrong with their compounds. And if you build a model based on this, then you might be doomed. So you really need to understand your data underneath and see if there is data shift in this thing. And typically what we do, we build two different types of models, classification and regression models, and maybe Alberto will talk about this to some extent as well. The nice thing about classification models, you can really nicely look at those bimodal data and predict this kind of thing. Regression model, the nice advantage of this is that you can actually provide a number to optimize against, right? That's very important for mostly the chemistry folks because they say, hey, is this more or better than this one? And if you cannot tell me, the model is useless. Yes, but if the data doesn't allow you to do that, then you shouldn't do it. What you want to have with models, they have to be predictive, robust, against new data, and you want to make sure that they're actionable, if possible, interpretable. I'm a person who nowadays says, I'm not sure if we really need the interpretation anymore. I know we always want to understand because we are scientists, but there are new methods, and I'll talk about one of them later, and I think Alberto would also touch on that one, where you can actually explore spaces without the need to actually come up with the ideas yourself anymore. Okay. The error part. This is an important one. If you have a model that works, and this is from a real data set, look at your outliers. The outliers might be that the model just doesn't know because it's discontinuous data, or you might actually have measurement errors. And we see this quite a bit. We've done this to some extent systematically, went back into models where we know they really work well across a wide set of data, look at the outliers, go back to the experimentalist, and very often we find that the data is actually wrong. So you can use your data, your models, to actually check your data, and that's an important point to keep in mind. Okay. This is not everything you want to know about data, but this is really the things you should keep in mind. Apart from the reality checks that you have, think about what are the models intended for? Can you actually use a regression model or not? Not everything is predictable, and specifically, the models will become invalid over time. So question when the model was built, what was the data underneath? And I think, especially for those people generating data, and that means also making compounds, design the data sets from the ground up. Do not just say like, oh, I just make the next compound because it will be the next active one, and I come to this in a second, how to maybe do this better. Okay. So much for the established methods. Now a little bit about the status quo and where we go. Some recent examples. I will not go into all of these. I would actually only touch mostly one of them. AlphaFold is a big thing, generative chemistry, retrosynthesis predictions, and also federated learning. And all of these, if you want, is really where I think where the field is going, what you need to keep in mind, and if you don't know about any of these, or only some of them, maybe have a look at what this is actually doing, because I think it's really a big thing. And all of these are moving very, very, very fast at the moment. Why they're moving fast is because it's often a mix of robotics and lab automation, AI/ML together with this, but then also using the data and sharing the data. That's a very important one, to make sure that the data that you generate as experimentalists are deposited in a central place and they're not living in Excel files or in PDF files or whatever you can come up with. Because the big advantage of machine learning is if we can go from hundreds to trillions of compounds, typically virtual compounds. And this is where then the next part of the presentation goes. I want to really focus on accessible spaces and why at large scale these things make sense, and also why maybe interpretation is not so important anymore. So you can access new spaces, chemical spaces, using reactions. So essentially something that is synthetically accessible. Something that you can do just by rules, and the folks from Roche have really have a nice thing with the chemist playbook there, which essentially takes things that chemists have done before, and you can reapply those things, and you can really access large spaces. And then the AI/ML-based things like genetic algorithms, autoencoders, so everything that falls into de novo and generative chemistry. But you can do more or less the same thing also with all the other entities. There are great papers out there if you want to do antibody generative design and things like this. Peptides, siRNAs is also slowly coming up there. So when you work with large spaces, apart from the established technologies, you can do large-scale ML predictions, really look at when is it expensive to generate new data or annotate your compounds with an endpoint, or you can also traverse the representation space very efficiently. So I want to look at these three topics briefly and maybe give you an insight of why they could be also really important in the anti-infective space, also with some examples. So first one, I pick here on David's profiles that he showed, because this is essentially doing the same thing just with targets. So what we've done here, this came from Eric, a presentation 2019, actually the work is ongoing for a long time now, is a system called PQSR. And what Eric and the team has built is that we have 8,500 Novartis assays that they picked. And what you can see, let me see, this is the pointer, no. Here on the left top side, it's a matrix of compounds versus assays. And you see this is the sparse matrix I was talking before. Building a model on this one is actually reasonably hard. It's doable, but what you do is you can actually build models for the single assays, the individual assays, to augment your data. So you inject, if you want, the predictions in the whole thing, and then you can use the full matrix for much better, much more robust prediction of if you would use the same profiles, but just on assays. And then you can predict this on new assays. And what Eric was able to show is that when we do this for 11,000 assays prospectively, if you want, with, it's not really prospective, but with a very stringent cross-validation, and you put a success threshold, which is at 0.3 external predictivity, which is roughly a 4-point IC50 measurement, then 75% of those assays are predictable. The same quality level as a 4-point IC50 experiment. And that, I think, gives you a good indication that you can actually understand not only in individual predictions, but you can do virtual screens by looking at similar things to such a profile. You can go in, and what Dave already said, you can do mode of action predictions, polypharmacology, off-target panels, and so on. So these things are super powerful. And with the new methodologies like generative chemistry, and I come to this in a second, they become even more powerful. Active learning is another one. So this is what I said before, important for those generating data. Really think about, because making a compound is not only time consuming, it's expensive. Really need to prioritize these things. So going always for the most active one is really a problem, especially for the machine learning part afterwards. So if you would be able to switch your mind and say, like, actually make the next data point in order to enable the model and not to find the next best compound, especially at the beginning of a project, that can be very important. So explorative, you can either do very balanced selections or greedy. And I try to show this here on the right-hand side. If you imagine the red line is the true profile that you want to see, and let's say higher is better on the y-axis. Typically, what people would go for is essentially find the points there at the top left, which are very high. The problem is that what you do with the machine learning model is the blue curve that you fit to this. And you need to get, especially where the gray zones is, this is your uncertainty, you need to get those closer to the red line. So if you only stick to the top left part, you might be actually missing some things which are down there in your low part, because maybe the error is not like this, but it could actually go up. So doing these kind of things, enabling machine learning models, really changing how you design your data sets from ground up is very important. And this was applied, not the same way, but very similar, in a large library screen for docking, active learning and docking. So essentially what's happening is that if you want to search trillions or billions of compounds, you cannot dock them all. Well, you can, but it's, it will kill some polar bears, so you don't want to do that. Essentially what you do is you take your compounds, you dock a set of them, you describe them with some descriptors in parallel, you put this all into a neural network, then you run this against a large database, take the either top X or a random set of your data set, you do 3 or 5 cycles of that, and then you do inference of the full data. So essentially you get a very cheap answer for your large data set that you want to work on. And here, Gentile et al., so the Cherkasov's group, has done this. They've used sync and real space, which overall is like 40 billion commercial compounds. They've done what they call deep docking. They have different strategies to do the hit selection, and in the end, out of the 1,700 compounds that they ordered, 1,200 or so were tested, and out of those, 117 compounds, which look really novel, were tested against SARS-CoV-2 with a, we can argue how potent this is, but they could show some potency. And I think this is really interesting because these are all just very easy to buy commercial compounds, typically $100 per compound. So that, I think, would allow us to find more diverse compounds for those targets and then go after that quickly. Which brings me to generative chemistry. So there they were looking at existing, if you want, or directly accessible from reaction compounds. With generative chemistry, we do something different. I will go over this quickly because Alberto will definitely go about this in more detail. So the idea is that you train a system to understand chemistry. If you want the grammar of chemistry, you project this into a so-called latent space. And then what you can do is you can take a compound, you project this into the space, and then using predictive models in this space, you can actually search the space to find better compounds, depending on what you say is better. So that's roughly how generative chemistry works in a nutshell. Different ways of doing this. You can do this with string-based methods, so just taking, if you want, language, like the large language models now as well. So ChatGPT actually does understand smiles. You can use graph-based methods. This is how we draw structures, so that's good. Typically, actually, string-based things work better than graph-based ones. And then you can actually also use these profiles that Dave was also talking about. So instead of saying like, hey, here's a structure, give me something else, another structure that's better, you can say, here's a profile that I want to have. So even if you want to say, with this profile, I don't care about the structure that goes in. This is the profile I want to have. Give me something that will show that profile. And I will give you an example on how that was applied on sugars. You can do this for exploration. You have something called distribution learning, which more or less you have a couple of compounds. You say, like, give me something which has a similar distribution. Or you do this exploitation, which is this traversal of the latent space. Okay, and this is how this works. It's PQSAR to compound. You already get the idea. PQSAR was there at the beginning, the profiles. And you come in with a profile and you say, okay, give me other structures. So how this works theoretically, now I would like to move, but I cannot. So I try this from here. You train your molecules with smiles, and essentially you do 2 things. You encode them into this latent space with the encoder, and at the same time you calculate the PQSAR models. And then you go with these things into a system which more is a discriminator. It's like a process where you show something to the system and say, like, hey, is this what I want? And then you also show some fake data from the outside. So you train it with the real thing, and you show something from the outside up to a point where the system cannot distinguish anymore. Is this a real molecule that I have, or is this something that comes from the outside which is fake? And then at some point you can say, like, hey, now you know what I want. This is the profile I want. This is where the inference phase comes in. Here's the profile I want. Give me something with some noise on top of it to get some diversity in, which has this profile. And then you can get compounds out at the end. I think this is super powerful, especially if you don't know what you are after. You only know what you want to achieve in the end. So Michal Pikusa and William applied this to sugars. They used a profile of 49 assays against different bugs. It was all cellular assay, well, actually not all, but mostly. They used a junction tree variational autoencoder for your generator. They've generated 100 compounds, out of which were 88 unique, and then they used a PQSAR model to do the selection. And, okay, good. What you can see here, I hope it becomes clear, is that essentially with the similarity plots, top at the left, you see that the further this is to the left, the more different are the compounds generated to what was there in the beginning. So you can generate very different compounds, both to the training set and then further below to public databases, also to the FDA-approved drugs. So you can really come up with something new that people have not seen before. And also the things at the top tell you essentially how drug-like are my compounds, and also the one at the top right, essentially how active are my things. So essentially you have everything. You want something new, you want something that is kind of drug-like, accessible synthetically, and also active. And this is what the things looked like. This is from their paper. So actually 3 compounds came out. One of them was unfortunately not synthetically detectable. Of course, that was the one that was predicted most active, as always. But for the other 2, they could show quite some activity, which was also along the lines of the predictive model, 1 to 5 micromolar roughly. And they could also show that there was no cytotoxicity measured. So I think this is really a method, even though it's still young and there is way to go. I mean, these things are not fantastic, but this is really something that does work. We also applied this in other spaces. What we learned is that you can really explore into novel spaces. The problem is that it also goes into spaces that are novel, which means that you show these things to chemists or even our internal systems, say like what is good and what is bad. People go like, oh my God, this is horrible, right? You see things like here, it's probably too small, but things with like 3 or 4 hydroxy groups. The thing is, our internal systems don't capture that because we've never made these compounds apart maybe from sugars, and they're filtered out with some other ways. So it works. You need the right team setup, and you need the right speed of this, but we are all putting this into place right now, and it looks, I think, quite promising, even though there's still way to go. Now, the thing about very briefly at the end, the thing about something that just doesn't look good, but we don't have it with the rules. How can you do this? And that was a research question that we had with an AI for Life resident. She was with us now for a year. She's looking for a job right now, so if anybody has a job, contact Ocean. She's really nice. So Jankin, as said, goes when no man has ever ventured before, if you want, right? And the question for us is that can we capture chemist's gut feeling about what is a good structure? You show a compound to a chemist, very often they know what is a good structure. I mean, it's just, they've seen so many of those, it's really fantastic. And the question is, can we do this? And what we did, we leveraged systems from chess, the ELO score, or from online gaming. So essentially, this is the matchmaking algorithm that sits behind the Xbox from TrueScale. We're working with Microsoft, so that was an obvious choice for us. So the idea is you have 2 players, they play against each other, one of them wins, and they play against, this person plays also against others. And then over time, you know which ones are roughly as good as the other. You do use this for matchmaking. So TrueScale has more things about how close are the people together, so the latency is okay with the computers and the networks, but for us, that's good enough. We did the same thing for molecules, and we really applied this now to generative approaches. The way this works is that we have a system, we showed 2 molecules to chemists, they say which one's better. Very generic question, there's some extra cases to it. We have a latent ranking model. Essentially, we do this in 5 rounds of active learning, and we have a reference data set, which is here in red. If you want, this is the stabilized, we asked multiple chemists the same question multiple times. And you see that with a couple of iterations, we really reach a limit, which I think will be actually also the limit, because, of course, there's an inconsistency in human priorities. And where chemists are really good, you see this, if you want, in the red part, you see where the chemists kind of agreed or disagreed, and the green part is where the model worked, and you see in the middle, this is where the chemists underneath each other do not agree. This is also where the model fails mostly. But that's okay. And when you're a chemist or into drug discovery, you see here we generated compounds only with that score, and you see on the left-hand side, we have attractive molecules. On the right-hand side, we have unattractive molecules, and I hope that most of you would agree that the left-hand ones look more interesting than the right-hand ones. And now we can use that model quite nicely if we use generative chemistry to say like, hey, you know what, all that stuff just goes out, and we take these pieces out. Actually, this thing is accessible as a model on ChemArchive. You just download it and use the model if you want. So come to conclusion, I hope I could show you that the established CAD and machine learning models together work really well. You can explore previously really inaccessible chemical spaces with these methods. You can generate them automatically. We need improved post-processing because we're also generating stuff that we've never seen before, so we need to handle that to some extent. And in the end, it all comes down to integrating these pieces with the project teams, because it's all new, we learn still, and it's not a golden bullet that will work for everything. And with that, I'm at the end, and thanks a lot for the attention.
Thank you, Nick. A lot of thoughts to consider. With this, I would like to invite Alberto Bresciani. He's Director of Project Biology at Exscientia. He has led or actively collaborated in multiple drug discovery projects spanning multiple therapeutic areas and modalities. At his company, he leads programs in the antiviral space and supports projects in malaria and tuberculosis. Please, Alberto.
Thank you, Ursula. So today I'm going to explain how the Exscientia AI platform works, and I will show you a couple of examples around anti-infectives. So first of all, let's talk about, thank you very much. So first of all, let's talk about the Exscientia vision. So at Exscientia, we believe that drug discovery and development are a learning process. So how does differentiate to standard way of thinking? So normally you have a disease problem, and then you have some underlying biologies that somehow explain that disease problem. And then on top of that, you build drug discovery programs. Like everything is fixed. But that's unfortunately not true, and that's the reason why a lot of things fail. What instead we think is that the drug discovery and development program must generate things to learn, additionally learn on the biology and on the disease project, so that we always work with multiple data and we always go back to the patient, having patient at the center, so that the project generate things that allow to best learn and so to find better treatment for the patient. And these things you will see permeate our entire framework. So this is a very complex slide, but and we don't need to go through each of these, and you don't necessarily have to read it. You can just look at me. The point is, drug discovery is a complex work, and we have to agree on that. And if you put AI on top of that, it becomes even more complex. So we cannot simply imagine that you start from a point, like there is a disease, there is underlying biology, you find drugs, and it's going to work. That's not working. Here we have a sort of embed cycle type of things. So it's not from one point to another, but again, it's constant learning. So we learn from the outcome, we learn from the clinical setting that feeds back into the biology and generate compounds at the beginning. Let's go through each stage one by one. And let's start from, targets. Right, so in anti-infectives, for example, in antivirals, if you are going for direct acting, you will have just a handful of targets, and that's it, that's easy. But if you start working on other pathogens, or you want to find human targets, for example, it becomes very complicated and it's hard. And choosing the wrong target at the beginning will affect a lot of investment in and put you at risk if you don't do the right thing. So how do we tackle that? Because Exscientia is an AI end-to-end, so we try to use AI machine learning in every step that we do. We have developed a platform that is called Center Biologist, and essentially this platform collects data at genome scales, collect public information, collects data that we generate ourselves in our precision medicine platform in Vienna, and then embed this information, create knowledge graphs, and then extract target information, targets with a higher probability of success. And that, I think we can all agree on that, but maybe the question is, and we ask ourselves the question, how can you prove that this works? And so we did a fairly easy exercise. So essentially, we took all public available information up to 2010, if I recall correctly, and used those information to predict which gene would have become interesting in the next 10 years. And then go back to the literature and see whether this gene did actually become interesting for and associated to the disease. And we were successful in about 40% of the predicted genes, which is a good amount for us, and potentially we were right on something that wasn't necessarily already discovered. So this is the first piece. When we have when we have selected what target and what area we want to work on, then we have to generate something. And this is where the AI machine learning bit comes into play. It's also at the beginning, but it's where I think it's more fascinating. So our precision design method is based on 4 different steps: initiate, generating the models, generative design, and active learning. And we've already heard a lot of interesting things around some of these. I will try to see if I can explain it in the way we do. Initiate is a very simple process. Everybody in pharma, when you have a project, need to set objectives. So objectives are set in the form of a target product profile. I'm sure this is not new to you. And then target product profile becomes target candidate profile, which is simply a list of features that you want, that are desired for your drug, associated to numbers, because you want to be quantitative, right? So we need to convert these PDF that cannot be used in something that can be ingested by our system and used. And how do we do that? We use scoring function. So we convert everything, every parameter, to a scoring function. It can be different for different parameters. And then these scoring functions are aggregated in what we call a merit score. A merit score simply could be even a very simple geometric mean of scoring function, or can be weighted. And the merit score tells us, the merit score tells us where we are. At early stages, we have low merit score because we are exploring, and as we move forward, we have higher merit score because we are exploiting. So we set the goals there, and we have the features we want for our compounds. But clearly, we won't be able to measure these features for all the compounds, for example, we design, because that's going to be impossible. So we need to create models to score against these features to, predict, the best compound. And how do we do models? I won't go, into the details. We've heard a lot of interesting things, important things on how a good model works. I will just stay higher level here and say simply that in general, you have 2 types of models: global models and local or project-specific models. Global models are things that are common to all projects, like, for example, ADME or PK. These things are not project-specific, so you build them using public information, previous project. You can also generate project ad hoc to learn and to create this model. And then you have project-specific model. There are, machine learning, deep learning system that are, models that are there for predicting, for example, potency, like single task, or multitask, like selectivity or something like that. So now we have our goals. We have our models, so we can predict against our goals. And now we need to generate something that will be used to predict something and check against the goal. How do we do that? How does generative design works at Exscientia? We have 2 approaches. One is what we call evolutionary design, and another one is design atom by atom. Evolutionary design is essentially taking some seed molecules and generating novel molecules with a system that knows what is possible and knows where the chemistry rule and chemistry modification were somehow encoded. And so this will create novel molecules by growing and merging and things like that, having in mind what was done before. And obviously, even in this case, you will get molecules simply impossible to synthesize. You will get molecules that are ugly, and we have filters, like it was shown, to get rid of these. The second part instead is building molecule one atom at a time. So it's completely unbiased and is based, it has learned what is possible in terms of linking the atoms, but that's it. And obviously it creates very fantasy type of molecules. Sometimes like you said, if you show them to a chemist, will probably pass out. But this said, there are ways to filter out, there are ways to find, and then these molecules are scored against the activity model, for example. So the generative design system learns what's best, and so keeps the connections that are best. And essentially, this is what happens. It's another tool that we have created that is called Center Chemist. So you have your seed molecules, you generate your molecules, then these are scored, filtered, synthetic attractability, desirability. They are filtered after model scoring in order to optimize multiple parameters, and you end up with a set of molecules. How many molecules you end up with after this cycle? Obviously, it depends on how many you start. But on average, in our case, it's in the order of thousands of molecules, or sometimes hundreds of molecules. But you cannot make thousands of molecules every design cycle. You cannot test thousands of molecules every design cycle because it's going to be too expensive ultimately. So how do we pick the compounds that we want to synthesize and we want to test? This is done through active learning, and we heard about before, and so hopefully you already know what it is. But in any case, active learning is a balance between exploration and exploitation. So let's think it this way. So we have the goals, the objectives that we set at the beginning with the parameters. Now we have a bunch of molecules with all the features, and we need a tool that tells us which molecule to pick to learn the most out of the set of molecules we have. So that we learn the most to update the model, and we learn the most to go in the desired direction with respect to our final product, right? So this is what active learning does, and essentially we give them the balance between exploration and exploitation. If we want to explore more, we'll unbalance toward exploration, so we'll cover the higher chemical space, and we'll pick compounds that are more different, but potentially not those that will have higher chance to be active. If we balance towards exploitation instead, we'll explore only some spots in the chemical space, but pick those that are more active, potentially more active, and trying to maximize the learning. So, all right, so now we have selected 20 compounds, and we are going out and synthesize those. What do we do next? We need to test, right? So, and the test is the final bit. So this slide essentially represents the entire ecosystem. So we have our objectives, we create models, we create molecules virtually, we score them, we pick the compounds through active learning to optimize, to learn the most in relationship to our goals. And then we need to do the assays that match what active learning has selected, right? So let's make an example. If I want to optimize against potency and permeability and whatever, I need to have potency and permeability and in my assay, because active learning picked the compound to optimize against those properties. So I need to be able to feed this measure back to learn the most out of it. And these are some implication in terms of how you do and how the testing cycle happens. So typically you have a stepwise type of things. So you first do your biochemical assay, and then your cell-based assay only on positive compounds, and then on nice compounds you do ADME, and then on super nice compound you do in vivo models or PK. Obviously it's not super sequential, but typically it's somehow sequential. Instead, in a AI-enabled type of program, to learn the most, you need to feed all data back into the system, because if you don't feed this data back, all data back, you won't learn it all. Obviously, sometimes it's impossible, so there are ways of learning even if you don't feed all data back, but in principle, you should be able to do that. And so the way you organize your laboratory is and your partner is dramatically different, because you need to have all these all together, not in a sequential fashion. All right, so I can probably skip this also because, we can go fast. It's just a sort of representation. It's obviously a fake, so where it shows some molecules and they are scored and they are tested. It's just to visually show how it works, but actually there is no true meaning. And maybe we can simply skip to more interesting things that are the two examples in the anti-infective area. So we thought, how do we exploit our system, so the potential of our system to tackle some of the biggest problems in the anti-infective? So, I think we can all agree that in the anti-infective area, something that we want to do is we want to kill fast. We want to kill our pathogen fast. That's the key to better efficacy. That's the key to low transmission. And that's the key to high barrier to resistance. So we want to kill them fast and we want to kill them quickly. So why we cannot explore polypharmacology in this fashion? So polypharmacology essentially means that I can target more than one thing, and if I can target two things in a bag, two completely different pathways, and I can get there with the right potency, then I can potentially kill fast as the consequences that I explained. And although this seems complicated on the right, it's simply another way of what I just told you. There are initial data on the bottom left, then you curate them, create the models, and then generate the compound, select the compound, test the compound. It's as easy as it is. It's not more complex than that. And let me show an example in malaria. So what we did, we gather all data that we could gather, publicly available data. We select them to pick those where we had more data quality was good, we had repeats that were optimal, and we ended up with 52 targets. Then we made a 52 by 52 matrix of pairs, and we generate compound through generative designs for all these pairs and score them against the activity model that were created with the foundation data, and filter them for desirability, synthetic tractability, and things like that. And then do the sort of butterfly plot in the beginning, and what you want to see is compounds like the yellow there that have some activity on both your target. On the x-axis you have one target, on the y-axis you have another target. This is predicted, right? So it's not already true. And out of that we selected 42, that's the Venn diagram there, that was then narrowed to 12, that was then narrowed to 4 based on key opinion leader inputs, and then out of these 4 we have one pair that is progressing through different multiple design cycles, and we are able to show that these compounds, they are active on a biochemical level on both targets. They do target engagement on both targets, and we see anti-malaria efficacy at the blood level with the activity that we predicted was there. And a similar thing is going on tuberculosis. We have 4 pairs, 2 targets, and we are at a stage where we we see antibacterial activity. Another example is more straightforward. Empro 1 year ago, we thought, why don't we? Because again, our system we can create this model, can potentially optimize against multiple parameters, and so we thought, why don't we, besides the COVID-19 goals, why don't we use this with the pandemic preparedness goal in mind? So we try to design something that is active, not necessarily only on MERS and SARS, that obviously everybody cares, but also is active on wildlife coronavirus with higher chance to spill over, so that we have something that is already there for potential future pandemics. And through some cycles of generative design and design make test cycles, we ended up with some compounds that are at least as potent as nilmatrivir, but they have a way better profile in terms of pan-coronavirus activity, especially in viruses that are not already infecting humans, but are like bat viruses, for example, high risk of spillover. Exscientia is building its antiviral portfolio around TB and malaria, as I said, but mostly around antivirus with a focus on coronavirus, flu, and paramyxoviruses. And we believe that this type of infectious disease are particularly suited for AI approach because, again, we can exploit, for example, polypharmacology and dual active compounds. And with that, I covered everything. Probably you were expected Denise instead of me, but she couldn't attend, so I jumped in last minute, and I hope I was anyway able to give you some flavor of what we do and a couple of examples on how we apply the AI to anti-infective therapeutics. Yeah.
Thank you very much, Alberto. Yeah, I forgot to say that you jumped in very late, and it was really a big thank you to you to come to this conference. So now I'm inviting all speakers for our discussion, and I also invite the audience to ask questions. I'm sure there are a lot of questions. I see already one hand. Please, could you start with your question?
I have two questions. One is a clinical question, and one is a non-clinical question. So for most projects, you're trying to separate two things, toxicity from activity, solubility, whatever it might be. But most of these things have very non-linear responses, and the center of non-linearity is often very different for the desired versus the undesired. And for example, fluoroquinolones, their activity, they're on 5 micromolar IC50, and then the other targets are 1 nanomolar. How does it relate to toxicity? Right, so how do you go about convincing yourselves that you have the right region of nonlinearity that allows you to really inform where to go? So that's the technical question. The clinical question is that some of the sources of failure of antibiotic treatment, which can lead to resistance, have nothing to do with the compound, not directly. They have to do with poor penetration into abscesses, into tissue compartments that are not well handled. And so how do you incorporate these sorts of issues, because these are one of the difficult ones for dealing with resistance?
Yeah, so maybe I can go, and then, I mean, if you hear me? Right. Okay, so first of all, with regards to linearity, you can have whatever scoring function you like. It could be also non-linear. It's not that you're forced to have, so when I said we convert our target candidate profile parameters into scoring function. So scoring function means it's a function that converts your outcome to 1, 0, and that scoring function can be whatever function. So if it's not linear, fine. If it's linear, fine. You can even make it a part linear and a part non-linear. You can do whatever you want as far as it makes sense, like you said. So you need to understand and optimize the scoring function. And it is a learning process, as I said. For example, in our case, we often start with some scoring function for the various parameters. And these are updated at some point because we've learned what is best, and our models have learned too. So you can use whatever function you want. And with respect to the clinical point, it's a very good point, but ultimately still a compound property. The fact that compound is able to get there, the fact that the compound is able to penetrate, right? So you can design compounds in order to get to certain exposure in a certain tissue, in a certain context, the best way possible. Then obviously there may be other things that you can do to facilitate that. You know, that's absolutely possible, you know, combinations and other things like that, potentially modeling that. But yeah, so you can in principle embed multiple desired outcome in the whole system, including you know, something that is not necessarily related to the outcome of an assay, like, you know, an IC50 or something like that.
I think we should use microphones. Yeah.
Yeah, somehow related to this question, one of the biggest challenges I see is that due to, let's say, challenging permeation and efflux, antibiotics are typically have to be dosed very high, and you need very high exposures. And for that reason, in my opinion, it takes sometimes many, many years in preclinical research. Many projects fail, starting with acute toxicity. And my question is, we can do off-target assays, then we can do something more phenotypic, or at the end of the day we do animal high-dose tolerability studies where these other assays have some limited predictability. What's your opinion on AI methods helping the researchers to increase chance of having good tolerability?
Yeah, I think these two questions, thank you for the question. I think it's a very valid one. And also coming back to commenting the first question, I think there are apparently two strategies. First, that you went deep into this area, spend as much as you can, characterize this nonlinear relationship, get as much as data points, train a model, and try to find the sweet spot. This is strategy number one. This is essentially a burnt ground strategy. The second strategy is you decide at the beginning whether you want to invest in this game or not. Now, always remember that this is the point I want to mention. Human intelligence is not limited to say we are forced to use artificial intelligence. So coming back to the question, this is a very good one. So one way one could improve the situation is to allow early screening assays that would improve essentially the predictivity, so that we don't waste a few years until we realize, this would have acute toxicity. And one other aspect, actually, very recently, you may have heard of this VX story, where people use generative chemistry to simulate or potentially design very toxic compounds. Actually, you can reuse the other way around. You can essentially, right? It's just turning the game. But combining these two, I want to, again, emphasize this point that we are not bound to use this. We use it whenever it makes sense. But it's important such considerations goes early into the development plan, so that we don't invest into unnecessary games.
Maybe I can just add one thing. So we've seen the profiles that Dave had shown, or the things that I've shown from PQSAR. So the experience that we have sometimes, it's not always the case, is that we only know about the off-targets too late. And I think that's really one of the issues that we had, at least when we didn't have some of those large-scale predictive models as well, that you would test against your standard assays or your off-target panels or whatever, and you don't even know that you would need to screen about something else in addition. And for those large-scale machine learning models, I think they can be really helpful in this space, especially if you have high exposure, which means in the end that even a 5 or 1 micromolar activity can be a problem. So I think those models can really indicate this. I didn't show it today. We had built a recommender system at some point, say like, hey, you have this compound, you actually want to look at that assay. I think that's another thing that, maybe more the AI for me was always more on augmented intelligence, so essentially warning the persons you should look at this. I think this is really another thing where it can really help researchers. It's typically not rolled out so much. We're much more used to this from Amazon or from Netflix or wherever, right? We tried it. It's really interesting. I think it's something that will come more and more together with digital twins and all these things as well.
Please here.
I was two questions going in the same direction. So all the data that I saw are more for looking into efficacy and safety, but I think what is also important to look into resistance. And how do you use AI to maybe predict if maybe some compounds will have developed faster resistance and some maybe have it will take longer. How can you do that with your models?
Yeah, so obviously, like we discussed, you cannot, so AI cannot, machine learning, so whatever, cannot invent something out of nothing, right? So, like it was presented, so it can predict, it can warn you what target could be of relevance because it has learned from existing information that potentially something could be of relevance to that. If there is no information there, obviously it's complicated to use AI to generate information that were not there in the first instance. So if regarding resistance, one can potentially, use not necessarily only AI type of approach, but additional approach to generate the seed information that can be used and brought to scale through the platform, but essentially is not a crystal ball, right? It cannot tell you something where there is nothing.
Can I just again add to this? So I think it depends also a little bit where the resistance comes from, right? There was, I cannot come up with a paper right now. There was a paper a while back where people looked with graph theory. When you think about a network of those pathways that we're talking about, and also thinking about compounds that attack two pathways or two targets at the same time, when you think about a standard network graph, you will always have some nodes which are kind of transferal nodes. Essentially, when you have one cluster, and then you have maybe one or two nodes that go from one larger cluster to another larger cluster. If you can identify where the resistance comes from, and you can specifically target those specific nodes that sit between clusters where there's essentially no mechanism by a bug to kind of go some other route to reach the same endpoint, you have a much higher chance to not hit resistance. And I think those graph networks very often can give you quite some information about what are the things you really want to target. So it can be also about identifying the right target, so that they are maybe less likely to end up in resistance. But if you have a single mutation, that might be more tricky to predict.
Another question would be then also, in one of the slides I saw there was patient is the best model, and I think this is the case because, they're not working under optimal conditions, you not have everything under control, and still the compound has to become potent enough to work and help to improve the conditions. How can you maybe build in also in your models, that even if you do not work with other optimal conditions, that you see that your compound it would be viable, than in the real world and help the patient?
Yeah, I believe this is for me. That's a very good question. So ultimately, the patient model, it's way more easy, for example, so in oncology rather than in infectious diseases, because you can somehow get some data and generate profiles, and then see which compound and which treatment and is best suited for certain type of things, and match this profile, and then create some sort of strong rationale for targeting something, targeting a certain patient's population against something that is best working for that. I think that in principle this is also applicable to infectious diseases, although it could be more complicated than that. But the data that were shown by Dave, for example, showing a single cell sequence coming from directly from a patient are going in that direction. So it's always a matter of having data that allows you to identify which type of population, in terms of human population, in terms of pathogen population, are best tackled by a certain compound or certain drug. This is a complex infrastructure, so complex, organization type of things, but ultimately the combination of these, so the clinician and the research side will definitely enable identifying the right population for a right target. Is precision medicine, is it possible on all these infectious diseases? I think it's more complicated than oncology, but potentially doable.
One thing that can also help with changing microphone is that, if you do not only predict your IC50s or whatever you have, but really try to build multi-target models, we heard this today, they can be very powerful, really to look at IC90s, IC50s, and maybe other things. So essentially K-ons, K-offs, and you model this all in one go. That can really help you identify the compound that's not obvious just with an IC50, which would also work with other ways if you want of interacting with the target.
Thanks. Bev Isherwood here from the Medicines Discovery Catapult. It's so great to see so many AI approaches being applied to infection projects. I guess what I'm curious about is whether any of you have a wish list of data sets that could really help you expand the application of these tools further in this field. It'd be great to hear your thoughts on that.
It's a fantastic question. So what would be the price tag first? Let me put this way. I start with the following statement, which can be totally bullshit, so bear with me. Biology is a complex adaptive system. It's complex, that means actually it has multiple pathways doing the same thing. It's an adaptive system, that means it adapts to new environments. And it's a system, that means actually it's not really let it reduce to a to single components to individually analyze it. That's why if I'm really investing in AI, let me put it this way, if I'm now, I want to do AI, definitely, I would like these two gentlemen focus on chemistry. Biology is really a damn difficult field to use AI because the system is so complex, we understand even not the surface. I think that would be my honest answer for that. As I said, though, we can discuss the price tag.
I think one thing that would be interesting, and we've tried that internally quite a bit, is now, and maybe that's just a small thing that you can think of, if you have more compounds around that were made and maybe you don't even test them or you can get hold of them from another lab or something in academia or in small companies, and you run these on plates and you have a couple of wells left, just chuck some extra compounds in because very often any data point will help. Eric had actually initialized that, something that we said every well counts. So if we have a plate with, I don't know, let's say 5 wells left, we would just inject other data or other compounds, stock solution that you have, and then we just get extra data because the wider you get, at least models will get more robust. So it's a small thing without a big price tag, I guess, if you have the compounds around.
Here, please.
So we've been talking about machine learning and we've been talking about the data required for machine learning, but something that we haven't spoken yet is how would you quantify the data, meaning how much of this data is needed for machine learning? And the second part is if you don't have that much data, does that mean that you should forget about machine learning?
I can start on this one. Okay, so briefly, I think obvious answer is it depends. So we've done some internal studies. If you want to use some of those, let's say, neural network approaches, a colleague of mine, Gregory, he looked at this with some interns and also others. And he said what you see is that you have really, when you look at learning curves, around 15,000 compounds when it comes to more or less whatever endpoint. This is where the models really reach a golden point where it kind of levels off for neural networks. Local project, you will never have that. So I think this is a rough guideline. If you say you want to use neural networks, if you can get to this point, this is often where you see an endpoint. And of course, you have large language models that will take whatever you have. I think that could be one guideline if you want. I don't think there's anywhere a point where you say like, there is no point because, as Alberto showed, even if you have only 3 data points, if you build a basic model and you say, where's my biggest uncertainty, and you pick the data sets, so you build them from ground up the right way for a machine learning model and not for what you think is the most active next compound, that's really where I think the focus should be.
I fully agree, and again, it goes back to the point I was trying to stress at the beginning, that is a continuous learning process. So it's not, you don't have to think about machine learning, AI, neural network, whatever, like something that is fixed there at the beginning and you simply somehow update. But it's more, a constant learning, constant adding data, improving the models, understanding whether not only the models, but whether the scoring function are working properly, and also understanding whether the reliability of your model outcome is good. For example, we typically have this system that is divided in quadrant that gives us, how a compound that is predicted through our models is good versus is reliable, right? So the bottom left corner is not good, it's not reliable, obviously you get rid of it. The top right corner is good and reliable, obviously you go for it. Then the top left corner, it's potentially good but low reliability, so nice to have. If I do not have any other option, I may consider it. Bottom right, it's high reliability but poor potential, then lower priority. So it's always a matter of balance. It's not just the outcome, but it's also, like we say, the error or the reliability associated with that outcome.
Thanks for the great session. I was curious, obviously you know that in CS, the open nature of AI has really pushed the field very much, and at least one of the speakers sort of talked to that. I'd like to hear from all of you about how do you do that in the context of drug discovery in a way that lets the field advance quickly, but of course still maintains what needs to be protected as well? Basically, how do we get to be more open in this field?
If I may, we at Roche, or many of us actually are strong believer of a sentence said by Eric Raymond. I don't know whether this name says anything. His one sentence is one famous sentence actually, given enough eyeballs, all bugs are shallow. That's why actually we open source the software that we create. For example, for molecular phenotyping, for the single cell data analysis, we created a package that we open source them. For sure, there are IP issues that we cannot simply release them without any further discussion, as well as, because it's actually also a lot of people's work. It's not that our side, but we try to stick to maximize the thing that we can publish. And especially and this is actually before the workshop, I think this is one of the value of this workshop, is to help us collectively think, how can we, that is my argument, we need more pre-competitive sharing of information, like the discussion that we had today, like the many good questions. How can we make this a regular practice rather than a privilege of a few people, including me here, to discuss with you? I think it's an open question for me.
First of all, 100% transparent is not my direct field, so I cannot comment about that. But certainly something that I feel from our tech team is that it's not one tool. It's not something. It's a combination of a number of tools. Some that are existing, some that you build, some that are free, some are commercial and they come with licenses, and so. If you have to rely on that bit, for example, to move something from one stage to another, then clearly it's not something that can necessarily be made available directly. So it's a combination, and this combination is not necessarily the same for all projects, and is not even necessarily the same within a project, but changes depending on the goals, for example, of every design cycle sometimes.
Yeah, and I think there's great things out there, like the Open Reaction Database, the Open Crystallography Database. There's also ways nowadays, if you're in a company, to share data. I think the Melody Consortium really has shown that nicely. If you're not aware of this, look at it. It's MELLODDEY. That, I think it was 13 pharma companies and some other academic labs. Essentially, it was the way you can share data really changes. So what happened there was essentially you build a machine learning, like a neural network, you cut a certain part off, and only that certain part that's cut off is passed between the different companies and kind of round-tripped a couple of times. And even though the predictions didn't get much better for most companies, the applicability became much better, right? And we see this now. And these kind of data you can actually then use to build meta models. And we will publish something, hopefully, if it's accepted soon, on how to use these things. So I think we will keep on open sourcing. We will keep on trying to share as much data as possible. A lot of what I've shown today is open sourced. So whoever wants to play with generative chemistry, I can just say, there's a lot of stuff out there, have a look. And I think we will try and keep more and more towards the open whatever databases.
Yeah, I think we are coming to an end with this field that's really moving fast. And maybe we can summarize this workshop with discovery is a learning process, and this should be shared openly. So with this, a big thank you to the speakers, and also a big thank you to the audience, and enjoy the rest of the conference.